We are moving straight to the next presenter, who is no other than Carolyn Angleton. She's bioartist and biotechnology researcher at SacBioArts. Now, Carolyn Angleton is a bioartist and biotechnology researcher. She has taught art and critical theory at California State University, Fresno, and Sierra College in Rocklin, California. She's co-founder of ArcBac, an art and synthetic biology collaborative at the American River College in Sacramento, California, and director of SacBio. She holds an MFA from Rhode Island School of Design and a BFA from Colorado State University in Fine Arts. She's also on the MIT Global Biosummit organizing committee and is part of the Community Bios Fellows program. Her sculptural and biological artwork has been exhibited as part of the Femme Gardens at the Ants Electronica exhibition in 2020, the Japan Media Arts Festival in 2018 in Tokyo, subsequently in Vembeka Foundation. Her exhibitions has been showcased at several international conferences and on several platforms. Her work has been shown throughout California at the Center for Arts, the Yaba Bernard Gardens in San Francisco, the Crocker Arts Medium in Sacramento, where I believe she schooled. She was awarded research grants from Biohack the Planet conference as part of the ArcBac's work on rendering a synthetic carotenoid pathway. And her work can be seen at carolynangleton.com. Without wasting much time, I welcome Carolyn to present to the audience. Thank you.
See my slide, and can you hear me okay?
Yes, we can hear you loud and clear.
Okay, and my slide, you can see my slide as well?
Yes, we can see your slide as well.
Wonderful, thank you so much. So my name's Carolyn Angleton, and I'm a bioartist and a biotechnology research. As Harry mentioned, I'm director of SacBioArts and a co-founder of ArcBac, which is the American River College Biotechnology and Art Collaboration. And I work alongside two microbiologists, Dr. Ken Kubo and Dr. Adam Talim. The title of my talk is Artistic Speculation and its Value to Science. One of my connections to today's conference is through my work on the organizing team for the MIT Biosummit and BioFellows program. It's through this experience that I know some of the organizers of this conference, and I'm very happy to be here today. The group of people in this picture represent something that's really important to me, that of building an international network of innovative, creative, social justice-oriented biotechnologists and biodesigners. In that sense, I appreciate that at a conference on biosecurity and biotechnology, that the idea of artistic creativity has been invited into the thought stream. It's my hope to broaden the notion of biosecurity to include the interconnected concept of biocultural creativity, where we see cultural workers alongside scientific workers as providing a blanket of protection through intellectual ideas, heartfelt actions, and accessible practices, ones that enfold our societies in a well-informed and a balanced sense of progress, and include a broad spectrum of voices here and now. I see bioart as a tool of envisionment that allows and trains people to practice forethought, to speculate on possible futures and their implications. So today what I'd like to do is to take you through a series of hybrid art science projects to show you a bit about how bioartists go about creating work. I'm not sure how familiar people are even of the concept of bioarts, but it's an artist who works with living materials. As a field of knowledge, conceptually based visual art is well versed in metaphorical content. Artists operate knowing that in depicting or creating new form, our thought process refers not only to the object itself, but a whole set of associations linked to it. And as such, an object becomes a symbolic departure towards new realities. As artists, we design around a set of strategic questions of embedded thought experiments. The word speculate has multiple meanings, as in invest in ventures or properties in the hopes of gain, but with the risk of loss, or to form a theory or conjecture about a subject without firm evidence. I view this as, where the field of biotechnology exists, as it tries to develop a roadmap, a progression as we go into an unknown future. I see my role as an artist as one who forms conjectures, risking loss, testing future scenarios, creating step-by-step hybrid art science protocols, which are tied together by a thought thread that exists over time and within multiple fields. I use my artwork to formulate a set of questions grounded in the practices and techniques of biotechnology. My work addresses how biological color is created. Color is something that applies obviously to art, and in chemistry and biotechnology, it's central to many bioassays and biosensors. The slides that follow are a brief look at a series of artistically based research projects attempting to understand and manipulate biological color. The first project up here is a work called Rendering a Biosynthetic Carotenoid Pathway. And one of the earlier speakers was talking about the production of beta carotene and developing new pathways for that. The carotenoid pathway is one found in nature and is well known in plants, algae, and bacteria. It consists of a set of genes that, when actively synthesized, can produce sequential gradients of color in E. coli. In our synthetic project, we constructed two plasmids, an arabinose-inducible one under the control of the promoter PBAD to express the gene CRT-EBI, the first steps in the carotenoid pathway, and the second one was a zinc-inducible one to express CRT-Y. We used Gibson assembly to join multiple fragments and transform them into E. coli. And then we will use gradient slants to produce a color spectrum. That's about where we were at before we got shut down by COVID. So kind of the way that an artist works, something like this, it works in terms of a metaphoric kind of basis where it cross-pollinates with two concepts. The first concept being the concept to render. To render is to cause, to be or become, represent or depict, translate and give back, process an animal to extract proteins or fats, as in rendering fat into lard. Pathway, a course of action, a way of achieving a specified result. A route, a path to walk down, a chain of cells along which impulses travel. The second project that we worked on, well known to iGEM participants, is manipulating color in GFP. GFP's molecular structure is that of a beta barrel concealing a chromophore in the center. Mutations in the chromophore cause spectral changes by changing the absorption wavelength. Through strategic single point mutations, one can create an incremental palette of color shifts. And the earliest ones of these that were done were green to blue to cyan to yellow. So what we did was in essence to recreate some of those early experiments, attempting to demonstrate that changes on a molecular level result in changes in a visual, perceptual, or aesthetic level. Part of our attempt is to use art as a way of exploring a concept outside of biotechnology labs is not something yet familiar to most people. Understanding that color, known to artists as a pigment-based material you can paint or draw with, that it can be produced by yeast cells reprogrammed to produce a protein of interest by providing new DNA instructions through inserting an engineered plasmid. This is something that's novel and unknown to the general public. Here we produce 2 liters of GFP through fermentation. For myself as an artist, learning these techniques is a way of expanding an artistic toolset and also redefining what could constitute an artistic endeavor, somewhere between lab-based bench practices and visualization of molecular products. One of the concepts I'm interested in with regards to color is variegation, which is the on-off production of color pigment. That's kind of the picture behind me, is a variegated petal on a microscopic level. I started doing research on camellia flowers, as they are a plant that frequently forms sports, which are genetically mutated branches that deviate from the parent plant. I began by making a visual library or herbarium of the progression of variegation through petals in individual flowers, which I gathered from a historic camellia grove that was near me in Sacramento, California. I was interested in the beauty of the patterns themselves and why people had so obsessively bred camellias for these characteristics. But I also wanted to know the mechanism behind why certain patterns were laid down and ultimately how to manipulate those patterns. Here's a couple of views of variegation. It's caused in 2 different ways. It can be, on the right here, caused by genetic mutation, in which case you see sharp delineation. And then on the left here, variegation can be virally induced by coming through the soil and the roots. And in that case, it creates more of these blotchy kind of patterns. The history of variegation research is really interesting. There's a famous horticulture researchers, Napoleon Jorgensen at UC Davis in the 90s, attempted an experiment where they were trying to insert an extra chalcone synthase gene into purple petunias in hopes of getting a darker flower. But it surprisingly resulted in knockout whites and variegated flowers. And this was a discovery that they termed co-suppression of genes before any of these mechanisms were really known. Their research in flower color mechanisms set the groundwork for Nobel Prize winning discoveries in 2006 by Craig Mello and Andrew Fire in terms of RNA interference, or RNAi, gene silencing by double-stranded RNA. So I think this is a good example of where kind of this visual looking into visual issues leads to some revolutionary discoveries that affected the whole field of synthetic biology and medicine and the discovery of retroviruses. So in our much more modest research with my microbiologist team, we began to look at ways to create sectors in bacteria, sectors being the growth pattern that creates variegation patterning. 2 different cell lines each start out as a single cell, then exponentially divide to create side-by-side triangular growth pattern. We started with Serratia marcescens, a bacteria known to naturally form red and white patterns. And by exposing it to different lengths of UV light, we tried to induce different rates of sectoring by inducing mutation. I don't think that we're necessarily successful in getting any kind of regulation on it, but it helped us in initiating a strategy of art making through a series of protocol-based experiments, which fits into a history of conceptual art practices in that it involves a set of written instructions aimed at achieving certain actions and results. We moved on to designing protocols to create sectoring and different combinations of bacteria species, designing around cell dilution, strain combination, aliquot size, and growth condition. Each step along the way of these related string of experiments is an attempt to get into more complex or sophisticated ways of looking at how to control and design with color patterning. The next step I'm aiming for is to create a genetic toggle switch or oscillator that could produce patterning through a bistable feedback loop. by using the LAC operon, which switches between a mutually inhibitory production of lactose and allolactose. One can create on-off pattern connected to reporter genes, or at least that's the idea of where I'd like to head with that. So that's a brief look at my research. If you want to hear more about these projects and details, I invite everybody to come to the Biosummit Art and Design track. Anybody involved in biotechnology is welcome to come to the Biosummit conference that will happen in November 19th through 21. And there, you just have to put in an application and express your interest in that. And it'd be great if some of a lot of the people here were interested in coming. There's tracks on all different subject matters, not just art. So to finish, I am so glad that this conference at SynBio Africa has gotten off the ground this year, and big congratulations to Odom and Harry and all the people who worked so diligently to put this together. It's really a tremendous achievement to do that this year in the midst of this conditions, and a wonderful step forward. And I thank you very much for having me as a speaker. Thanks.